Good morning to all delegates from India. Welcome to this K Mohanty Memorial Oration. The Department of Pharmacology of AIIMS Bhubaneswar is organizing this oration in clinical pharmacology since 2020. This is basically giving a tribute to renowned pharmacologist of late Professor K Mohanty in the second year of this oration. We are thankful to Dr. Deepika Mohanty, who is the daughter, who has joined us now. Is a consultant hematologist and the daughter of Professor K Mohanty to initiate the support of this oration. This year, we are glad to have Professor Abdel-Rahman with us to deliver her talk on moving beyond useful, translating pharmaco knowledge into usable decision support. This is something very interesting. And we are doing research, but what is the meaning and what is the contribution to the mankind? Probably that is something interesting to learn from her. Professor Susan is a clinical pharmacologist and professor of pediatrics, University of Missouri, Kansas City, USA. We know it little odd timing in Kansas City. It is night, and thank you, Sue, for waiting for us because it is morning. It is just 9 morning. Thank you again for Professor Susan for joining with us. Now I request our director, Dr. Gitanjali, to give her opening remarks. Professor Gitanjali, please.
Thank you. Thank you, Professor Hota, Dr. Hota, our Dean Academic, Dr. Deepika Mohanty, and Professor Susan Abdel-Rahman, a good friend of mine, whom I am meeting online virtually after I think more than a decade. And it is indeed a privilege that Professor Susan Abdel-Rahman has agreed to our request to give this oration at AIIMS Bhubaneswar, even though we are meeting virtually, because this is a very prestigious oration. We are honoring a very well-known clinical pharmacologist of India who has done phenomenal work in India, and this is being done as a tribute to him by his daughter, Professor Deepika Mohanty, who herself is an immunologist of repute. And so it is really very heartening that we clinical pharmacologists of India as well as abroad come together to listen to this oration because it is an extremely prestigious one. And I am really happy that Susan has agreed to talk on this wonderful topic. The first oration was given by Professor Crisanta Veerasurya, who was with WHO in Geneva, as well as he was also working in New Delhi at CRO. And this is the second oration, and I am honored to have a lady this talk, as well as to be able to listen to Susan. I listened to Susan maybe a decade and a half ago when she gave a talk to the residents of Sishu Bhavan in Cuttack. And I was so enamored with her talk that I have been wanting to have her give this talk. And when this oration came around, there was no doubt in my mind whom I wanted to select for this thing. So thank you very much for accepting our invitation, Susan. And over to you, Rituparna, to conduct the day's proceedings. Thank you very much.
Now I request my colleague, Dr. Rituparna, to deliver a brief presentation on the professional achievements and research works of Professor K Mohanty.
A very good morning to all. It is my proud privilege to present the biography and the professional achievements of Professor K Mohanty, who was an eminent pharmacologist and also a renowned physician from the state of Odisha. Dr. K Mohanty, he was born on 1st October 1909 at Balipadia in Cuttack district. He passed his medical examination from Odisha Medical School with gold medal and 9 silver medals in different subjects. He was awarded LNP diploma in 1936. We all know that in 1930s, that time LNP diploma was the license to go for medical practice. After that, he joined as a medical officer in government of Odisha, and in next 10 years, he served different parts of Odisha, including very interior areas of Koraput district. During this period, he had many difficulties, especially his patients who were mostly poor and illiterate, and they are highly suspicious about the Western medicine because we are talking about pre-independence era. And he often had to win them over to accept him as their well-wisher to bring about wellness in their lives. In next 2 years, from 1946 to 1948, he completed a condensed MBBS course from SCB Medical College, Cuttack. After that, he was appointed as demonstrator in the Department of Pharmacology in 1948, and later he was promoted to assistant professor, and he was also the physician in charge of therapeutic ward of the teaching hospital. In 1952, he was awarded the Commonwealth Fellowship under Colombo Plan. And under this fellowship, he joined the University of New Zealand. In 1954, he completed MD in Pharmacology and Therapeutics from University of New Zealand, Dunedin. These are some of the photographs of Professor Mohanty with his professors and colleagues during his days at New Zealand University, Dunedin. Because of his bright academic career and his passionate interest in research, the university offered him to join the Faculty of Medicine, and he joined the Faculty of Medicine, Otago University Medical School, New Zealand, as associate professor. But in the same year, he changed his mind and returned to India and rejoined his post in the Department of Pharmacology at SCB Medical College, Cuttack, and 4 hours after, he was promoted to professor in 1958. Later, he was transferred to VSS Medical College, Bulla in 1960. He was also ex officio head of the School of Pharmacy from 1962 to 1968. He retired in 1968. Right from the beginning of his professional medical career in 1930s, he developed a passionate interest in clinical and laboratory investigations and research. Despite multiple limitations and with no basic facilities, he started thinking, investigating, and researching ways to treat and heal patients in his improvised lab. And he has published his important works in very international peer-reviewed journals like Nature, Antiseptic, British Journal of Pharmacology, Journal of Pharmacy and Pharmacology of London, Indian Medical Gazette, Journal of the Indian Medical Association, etc. Among his important works, the studies on sex hormone, sperm test of pregnancy utilizing male toad Bufo melanostictus was published in Indian Medical Gazette in 1950. Later, it was referenced in the textbook of J.P. Modi's Medical Jurisprudence and Toxicology and Physiology of Sex and Reproduction, published by Edinburgh University. Another path-breaking work of Professor Mohanty was a new method of assay of human chorionic gonadotrophin utilizing male toad Bufo melanostictus, which was published in the Journal of Pharmacy and Pharmacology in 1951. His work on development of tolerance to ganglion blockade by methonium compounds was published in Nature in 1954. The continuation of the same work, he published the development of tolerance and cross-tolerance to methonium compounds in laboratory animals and was published in British Journal of Pharmacology in 1955. Later, it was cited by Sir F.H. Smith in his book, High Arterial Pressure, which was published by Blackwell Scientific Publisher. During his days at Government Headquarters Hospital, Koraput in 1940s, he published few important case series and case reports in the Indian Medical Gazette. For example, Mepacryl intoxication and vitamin B deficiency and maternal blood in whooping cough were two very important case series published in 1945. In the same year, he published two case reports like the presence of rhinoscleroma in Koraput district of southern Orissa and the occurrence of leptospirosis. The occurrence of leptospirosis. It was published in the Indian Medical Gazette in 1945. Another important publication, Atebrin ban of amyl actridine derivative in human malaria, was published in Antiseptic in 1945, and it faced him a very prestigious Winthrop Award. His contribution, especially in the field of ganglionic transmission, has been cited in the recently published which has been edited by Dmitry A. Kharkevich. Not only contribution to science, his contribution to Odia literature is also worth mentioning. He pioneered and prepared the English Odia medical terminology entitled Ayurvigyan Paribhasha Avidhana. He also made his mark in the field of Odia literature and wrote poems, novels, essays, and plays. Boitojatri is a novel for children. Sishuro Vikas, a collection of essays. Pratik Dhwanira Dhwani, it is a collection of poems. Sagar Sangam, it is a novel. Rajasundari, it is a play. Sagarika O Ananda Galpa, it is a short stories. And Vishesh Patha, it is a book on nutrition. Among these, the book Boitojatri received prizes on children's literature, both from the Government of India and Government of Odisha. Later, the book Boitojatri was translated to English with the title Three Mariners, which was published in 1993. Professor Mohanty left us on December 5th, 1994. His immortal works, his blessings are with us, and this particular oration is a tribute from our side to a great legend, Professor K Mohanty. Thank you all. Now I request Dr. Vishwamohan Padi, Additional Professor in Pharmacology, to introduce our esteemed speaker, Professor Susan, to our audience. Dr. Vishwamohan, please. Presentation.
Good morning to all, and good evening to Professor Abdel-Rahman. It's my privilege to introduce Professor Susan M. Abdel-Rahman, eminent clinical pharmacologist and professor of pediatrics at the University of Missouri, Kansas City, USA, as the distinguished speaker for Professor K Mohanty Memorial Oration 2021. Professor Abdel-Rahman is a highly accomplished researcher, innovator, leader, educator, and mentor with a proven track record in the development of drugs, devices, and medical software. After obtaining Bachelor of Science in Pharmacy and Doctor of Pharmacy from Rutgers University and postdoctoral fellowship from the Ohio State University, she has been actively involved in teaching, research, and patient care, and is currently holding multiple senior positions. Since 1998, she has been working as a pediatric clinical pharmacologist in Children's Mercy Hospital, Kansas City. She also holds the prestigious Marion Merrill Dow Missouri Endowed Chair as Professor of Pediatrics at the University of Missouri at Kansas City. In this capacity, she has maintained an active research program which has produced more than 150 peer-reviewed publications, 54 grants, 30 technical reports, and 2 white papers. She has extensive experience in research project and personnel management and has enrolled over more than 28,000 patients as principal investigator, largely in community-based. She is also involved in didactic teaching and practical training of physicians, nurses, pharmacists, and undergraduate students, and serves on various institutional boards. Professor Abdel-Rahman serves as the Chief of Section of Therapeutic Innovation, Division of Clinical Pharmacology, Toxicology, and Therapeutic Innovation at Children's Mercy Hospital, where she and her team run an independent program for creation of novel drug formulations, development and implementation of pharmacology-based clinical decision support software, inventions, patents, and regulatory clearance and licensing of medical devices. In addition, she also provides her technical expertise to various reputed pharmaceutical firms, including Abbott, Merck, Eli Lilly, and Santeros. Since 2018, she is also serving as the Director, Healthcare Innovation at the Children's Mercy Research Institute, where she is involved in strategic and tactical planning and policy development to promote healthcare innovations. In this capacity, she oversees the preparation and submission of patent applications, external partnering agreements, and other regulatory documents. She has many innovations to her credit, such as the MUAC Z-score tape, electronic health record integrated precision therapeutic software, and pediatric weight estimate device. Her scientific contributions have been well recognized, and she has been bestowed with numerous awards, including the CMH Department of Pediatric Mentoring Achievement Award, the CMH Department of Pediatrics Entrepreneurial Achievement Award, and Golden Apple Mercy Mentor Award, and many others. Professor Abdel-Rahman serves as a technical advisor to various reputed scientific organizations, including the World Health Organization, the Critical Path Institute, and Institute of Medicine. She is an important functionary of American Society of Clinical Pharmacology and Therapeutics, and was the president of the society in 2020. She is also an active member of the Missouri Department of Social Services. So apart from her stellar academic and professional achievements, she is also actively engaged in community service and is involved with the Harvesters Food Bank, Meals on Wheels, and Parkinson's Foundation of Heartland, and many other such organizations. So with this brief introduction of our eminent speaker, may I request Professor Abdel-Rahman to kindly deliver the oration.
Thank you so very much. Let me go ahead and share my screen. And I will ask you to let me know if you can see the presentation.
Yes, ma'am.
Yes, we can see it. Yeah.
Okay, perfect. Well, it is really a distinct pleasure to join you today to deliver the Dr. K Mohanty Memorial Oration in Clinical Pharmacology. I have such lovely memories of visiting Orissa with your esteemed Dr. Gitanjali, and couldn't be more delighted to join you today unless I was there with you in person. I would like to spend our time together talking about one strategy by which we can disseminate the important knowledge that we create and possess as researchers and practitioners in pharmacology and the pharmaceutical sciences. I hope to bring a slightly different perspective to how we can approach the broader goals we may have for the work we perform in these fields. And I will conclude with a description of the lessons that we have learned and how they align with the career of Dr. J.K. Mohanty. Before we start, I'm going to ask you to take an imaginary journey with me today. We will be departing Orissa, maybe from the Biju Patnaik Airport, for a pharmacology meeting in Chicago. En route, we get delayed at a layover in Istanbul, so we set out to find some seating on which we can comfortably do some work from our laptops. As we walk through the airport, we come across 4 seating options. Using the chat feature in our Zoom platform, I'm going to ask you to share your opinion of the chair you would most like to sit in. And I am going to see if I cannot see the chat feature. So I'm going to ask Dr. Gitanjali to tell me what she's seeing, what people are choosing for the chair that they would like.
I'm seeing C, mostly C's and 2 D's.
Okay.
D's. C. Mostly C. Everyone is choosing C, including myself.
Thank you, everybody, for sharing your opinion. So looking at these 4 options, we can argue that they are all useful. And by useful, I mean that they solve the problem of providing us a place to sit down. But I would argue that they are not all usable. And by usable, I mean intuitive and functional. I'm not sure how I would even approach sitting in chair B, and I'm not optimistic I could sort out how to work on my laptop from chair D. Now, my instinct is that only chair C would be desirable for a lengthy layover, but my husband and some of you in the audience point out that you would enjoy sitting in chair D. So maybe 2 of these chairs meet the criteria for being desirable, meaning that you prefer this experience over others. And we'll get back to the importance of usability and desirability in a minute. In actuality, we are all on a journey together, albeit a slightly different journey. We all operate or are training to operate in the what I'm calling the pharmaco space. It may be pharmacology, it may be pharmacometrics, it may be pharmacoepidemiology, or any of the other pharmaco disciplines. And I suspect that many of us chose this path because at some level, we want to improve the use of medicines in patients, to use them more intelligently when and how they are needed in individuals who will benefit, while avoiding them in patients where they will do more harm than good. This is the essence of precision therapeutics. For me, the accompanying challenge is the ability to integrate my knowledge into clinical practice, but for you, it may be drug development or regulatory decision-making. Each of you is generating knowledge that informs how to best use medicines, but apart from downloading your consciousness to your end user, the path to practical translation for your work may not be straightforward. And that is where decision support solutions have a role. So today I'm going to talk about our experience creating decision support tools for prescribers and the various approaches that we've undertaken to do this in the clinical environment. Our process aligns closely with the best practices that are laid out by a healthcare quality agency in the United States. And this is an older reference, but still useful if you're looking to understand the basic principles of creating solutions that are usable and desirable. I'm a practicing clinical pharmacologist, and the decision support I develop is largely driven by the consultation requests that I get in practice. I know how to solve the problem in a way that's most efficient for me because I do it for a living. But that doesn't mean that I solve the problem in a way that makes the most sense for the providers. So the most fundamental thing that we do when we develop decision support is to co-develop these tools with the local users. Engineers are very smart people, But having spent a lifetime around them, I know that left to themselves, they will develop solutions to impress other engineers, not necessarily the users of the technology or the solutions that we're building. In fact, these are all the people that we engage when we're building our decision support. We have a core team that represents pharmacology. If there are laboratory methods that are being used in the decision support, our laboratory medicine team is involved. There may be other clinical subspecialties like hematology oncology. And then we've got peripheral involvement from nursing and pharmacy and information systems and senior administration and even business development. And I'll explain their role in a minute. These tools should be available at the time and location of decision making, which for my purposes means being integrated into the electronic health record. The value of these solutions diminishes when my prescriber must step away from his patient and the system in which he normally works to try to get to an answer with my solution. The tools should transparently pull necessary and relevant information so that no additional data entry is required. As you can imagine, this is not a trivial exercise. What you see here is an example of the first piece of code that we wrote to extract the patient's weight from the electronic health record. Well, it turns out when we ran this code that it returned 356 independent fields that all referenced weight. So we had to more carefully design and craft our code. Some levels of evidence should also be available for the user to explain how you arrived at the solution. And the recommendation that it provides should be clear. A recommendation to reduce the dose is not very helpful. A more concrete recommendation to reduce the dose by 75% is much easier for my users to implement. Our process starts with a requirements analysis to fully understand the needs of the various individuals that will interact with the system. And we go back to these users with a wireframe before we even begin to prototype. So we show them what it would look like before we start the build and make sure that it makes sense to them and their workflow. We also conduct very careful unit and integration testing, which means confirming that each element of the software performs correctly alone and when combined with other elements. So depending on the complexity of our algorithm, we may run 10 to 30,000 mock data sets through our code and examine the results against solutions that are independently coded by another application like MATLAB. Iterative improvements are made in response to functional testing. And while we focus on the end user, we actually also engage other providers who are less familiar with our objective. They are more inclined to randomly explore elements of the interface that we hadn't considered. In essence, almost stress testing the system for us. And as you can expect, the work does not stop once the product is released. There is continual regression testing that is done and refactoring as we identify bugs and encounter edge cases that want to crash the system. Development in a clinical environment looks very different than you would expect if we were part of a design-focused company or a software company. And that can be a good thing and a bad thing. We have direct access to the problem and the users, and that is a big advantage. But the facility with which we can develop the technology is a whole other story. And I've seen this play out in several different ways, and I think there's some value in sharing all of that with you. So we've got strategies where we engage the traditional information technology group in our institution. And the benefits of that is we develop compliance systems that are fully integrated with the organization. However, the job of most of our IT professionals is not to develop these type of tools. And so we're asking them to wear a second hat that they don't normally wear when we co-develop with them. And because of that, they are often pulled away to do mission critical things for the organization. There are communication gaps. We don't always speak the same language, and there's a fair bit of resource costs in pulling them away from what they do. And for this reason, many people will develop shadow IT, which means they just kind of build the software on their own in the background. Now, this is a very efficient way to build the system, the software, but it's not compliant and it's not integrated with the system. So it stands alone and it's much more difficult to access. And then more recently, we have specific IT innovation streams within our organization that I've helped to build. And these are ideal because not only are they compliant with the organization, but they're more streamlined than working just with IT alone. And they've got dedicated personnel to the project. And I'm going to share examples of each of these with you and the positives and the negatives. So our first example is software that we built for optimizing the medication dosing of a drug called busulfan. And busulfan is a medicine that's used in oncology when somebody is preparing for bone marrow transplant. Getting too little of this drug could mean that you fail to engraft the bone marrow that's being transplanted, and getting too much of this drug could damage the liver. Both of these problems can cause death of the patient. And the problem is that there's large inter-individual variability, meaning I can give the same dose to two otherwise similar individuals and the concentrations in their body will be very, very different. So exposure-guided therapy is standard of care, and it's even in the regulatory approval in the United States for the administration of this drug. However, we have a few issues. One of the problems is it's based on laboratory testing that has in the past for us required an external laboratory that had to be contacted more than 48 hours in advance. We could only administer the medications Monday through Thursday because getting the assistance of the outside laboratory and the additional support we needed wasn't available on the weekend. There was a lot of manual logging of information and samples. There was a lot of coordination of calling back and forth, so offline activity to transmit information. And it took more than 24 hours between the time we first started dosing a patient and the time that we could make an adjustment. And there were additional problems. This was the type of recommendation we were getting from the lab. If you can see the circles, these are the patient's data. And if you look at the dark line, this is what the laboratory was telling us they fit the mathematical data to. Now, anybody looking at that can tell that that's not a strong mathematical fit of those data. And I would argue that this is analogous to trying to predict how fast the skier is going to ski the slalom by estimating their speed straight through the path without taking the appropriate route. And so when we compared the solution we were getting from this lab to our own more robust model, we found differences in dosing recommendation of as much as 30%. So we created a decision support tool, and I hope this video will play for you, and I will talk you through it as we start to play. But you can see that this is a system that's automated. It's integrated into our EHR. You can actually load it from the patient's profile or go in with their own separate electronic medical record. It asks you to confirm that this is the patient that you're caring for and want to treat. And then it pulls all of the information that's necessary for decision making. It pulls laboratory data. It pulls data on dosing from the medical administration record. It pulls nursing data that tells how the drug was administered and how the samples were collected. And then it provides the information in both graphical and tabular form. It gives the user options of how they want to do the mathematical modeling. And you can see it tells them what was done and provides several different ways to check how robust the mathematical modeling was compared to the actual patient's data. Some of it is in tabular form, some of it is in graphical form. And if the user wants more information on how we came to look at this reliability, they can click on additional tools and take a look there. then they can actually simulate. They can see where the current targets are right now. And these were designed specifically for the users, in this case, our oncologists who are doing the bone marrow transplant. They know they're looking for a specific target, and they can go ahead and identify where the target is now and where they want it to be, and what the corresponding dose that's needed for them to get to that particular target. If the dose is not an even number, in this case 51.9 milligrams, they can go back in and readjust the dose and take a look at what happens when they round the dose up or down. And the providers can iterate like this as much as they want before making a final decision and pushing that recommendation back into the chart. And the nice thing about this is that it all can be done online without ever making another kind of step with the patient, and they can explore exactly where they want to be. So the software actually walks them through reporting, and the reporting actually pushes directly back into the electronic medical record. And then the recommendation actually pushes back into the ordering system to allow the user to make sure that the next dose is ready to go. And it should be running its course. So you can see in this case, the user can make some comments. They can update the report. They can add their own little notes that explain why they were doing what they were doing and push it right back into the system. And I'm going to get started. So not only do we create these tools, we don't just launch them and then leave them. We carefully evaluate human factors and usability by looking at system quality and asking our users how they feel about that, looking at information quality, and then looking at how they like or dislike the overall interface and the overall system itself. And there are a few things that we got right when we developed this, and there are some things that didn't work so well that we got wrong. The first thing is that our physicians embraced the product. They loved it. They used it immediately. It was also used to expand revenue for the organization. So not only could they deliver this type of precision dosing to their own patients, they could now deliver it to patients at other local hospitals that were using the medicine but didn't have access to this type of technology. However, it took us a very long time to build because we were trying to do it with the hospital IT system. And we had to refactor or recode this software 3 times to make sure that it could integrate with the electronic health record. So while we have a product that is still used to this day, it wasn't very efficient as we demonstrated, as we developed the product. So here's an example of another solution that was built the completely opposite way, that was just built using shadow IT and no connection to our hospital system. And in this case, this was more of a research application. There's a drug called atomoxetine that's used in children for attention deficit disorder. And in this case, our partners were the developmental and behavioral medicines. It also has very large inter-individual variability and mixed responses in patients. And so I'm going to show you some data that we generated at our organization. This is the medicine given to children, and these children are stratified by their genetic code, so how their body indicates they should break down this medicine. Now, all of these children were given the exact same milligram per kilogram dose, but you can see that there's a 50-fold difference in exposure between the child with the highest concentration and the child with the lowest concentration, and this is after the exact same dose. So if our researchers in developmental and behavioral medicine want to understand why children respond differently to this medicine, they won't know whether it's because the child inherently has a predisposition that wires them up to respond or not respond, or it's because they simply didn't achieve the concentrations that were necessary. So this group wanted to do research where everybody had the same exposure. And if everybody has the same exposure, then we can tell whether or not they're not responding because of some genetic predisposition, or they're not responding because it's not going to be affected because it's an exposure issue. They knew what target they wanted to hit. As you can see here, this is response data that was generated in a separate research study, and they knew that effect plateaued somewhere between 400 and 800 nanogram per mil. So if they could find the right dose to get to this concentration or exposure in everybody, then they could really start to research the problem. And what we did was developed a solution that's far less sophisticated. I'm not going to walk you through it here with a video, but what it does is requires the user to put some specific patient variables in, and then it tells them the dose that they need to get to their specific exposure. So it was a very clean and quick, but not very sophisticated decision support tool. And we did have some things that we got right and some things we got wrong as well. It took us only 3 weeks to build this software, and it actually worked very well. We were able to reduce inter-individual variability from 50-fold to 6-fold, and that meant we could rapidly iterate on this software. But it's got limited application outside of the group that's using it because it's not connected to anything. It's just a standalone piece of software. So while it works effectively for its cause, it's not ideal for much broader application or dissemination of this particular knowledge. Now I'm going to share a third example that we built using an IT innovation stream. So here we take IT experts and pair them with researchers and scientists or clinicians in a role that specifically has them dedicated to building this type of software, to supporting innovation, so that we don't have competing priorities and we can really move much faster. And this particular software that I'm going to show you was built to address an issue with the anti-hemophilic factors or the blood clotting factors. Our partners here were hematology, and there were a couple of motivations for building this tool. The first is that these are very expensive medications. They cost our patients in the United States about $30,000 a month. We also have very mixed adherence rates. So we see adherence or compliance rates that range from 6% to 160%. I mean, this is really almost unheard of in most other conditions. And we've got very wide inter-individual variability as well. There was also another unique caveat to this particular drug. Our hematologists were used to doing dose optimization and for their hematology patients with classical pharmacokinetic approach that I'm going to show you. But in the more recent literature, people were moving to a population pharmacokinetic approach. So not only did we want to provide them a tool that would allow them to do what they were comfortable doing, but we also wanted to give them the option to explore whether this new approach that others were investigating or experimenting with, population PK, would be useful for them as well. And we built this tool. It's not a video, but I'm going to show you some screenshots that look very similar. You identify your patient with their electronic medical record. You can find the patient and the instance that you want to do the curve fitting on. You'll see the exact same thing we saw with the oncology application, where we pull in medical administration or pharmacy data, MAR data, nursing data, laboratory data, all in one space to examine. We give the provider the fitting and show them how well the modeling works, and then show them how well various population models work as well in their particular patient population. And then we give them options for identifying the right dose when the patients come into the hospital for an acute bleed and when they come or need prophylaxis on a regular basis. So it's a little bit more complicated than our busulfan tool, but again, answering the questions that our providers were interested in. We also had an additional objective here. And this is our reporting feature as well. Again, we did the same human factors testing after we developed it, but we had one additional idea when we built this, and that was to extend the application to the children and their parents. I mentioned a few slides ago about adherence for this medication. And the reality that we were dealing with adherence that ranged from 60 to 160% didn't sit well with us. We wanted to promote a shared decision-making tool that would inform the patient about the decisions their doctor had made and allow them to understand what happened when they chose to make changes to their own regimen. And here I'm going to show you, I'm going to walk you through what it does. So the family enters through a patient portal that we have. They can look at the recommendation the doctor has made. They can understand what that recommendation translates to from a standpoint of how much factor is left in their body at any point in time. So what is their risk a day after the injection and 2 days after the injection? I don't know if this is going to move, how quickly this is going to move, but you could see as they toggle through, they can watch what happens to the change in how much clotting factor they've got left on board. A fun little feature, the little child you see there, they can actually design their own avatar so that we can personalize the solution for them. And then they can go in and explore what happens if they are not comfortable with this regimen. This every 72-hour regimen is great from a pharmacology perspective, but that translates into dosing that is Monday to Thursday to Sunday to Wednesday. It can be a little hard to follow. And so we had happened to notice that our patients that were getting recommendations like this were just saying, well, what if I just give it twice a week? What happens? And so what they can do is actually say, I want to go in and see what it looks like when I give it twice a week. And the goal isn't for them to necessarily find a better dosing recommendation, but the goal for them is to really understand what happens when they choose to make changes that their doctor didn't recommend, and to hopefully promote a better conversation with their doctor. And again, we've done very similar testing with this tool. We've gotten a lot of feedback from our users. You can still, this is still going through different variations that the user can play with as well. And I'm gonna go ahead and move through that. It's walking through now an example of what happens if they choose to change the dose, and they can still get a sense of where they are as well. So what did we get right in the development of these tools? Well, they were built certainly a lot faster than tools from the traditional IT stream, a little slower than our shadow IT, but that's okay because these are fully integrated within our organization. We were able to expand patient engagement, and we were able to allow our investigators to explore new clinical strategies and perhaps do a little empiric research when they were caring for their patients. It still requires our families to go in and externally authenticate, so it means they have to have a password, which is not ideal. It's not a one-click to get into the application for them, but it's still a very useful tool. And so there are a number of sources of success that we've had. And what has made us successful are the same things that made Dr. K Mohanty so successful over the course of his career. Our teams welcome these resources because we had developed a foundation of trust and credibility. Much in the same way that Dr. Mohanty early in his career had to win over his patients, who we heard today were poor and suspicious, so that he could introduce wellness into their lives. These tools that we created extend our knowledge and empower other providers to integrate this knowledge into their practice. And in this same way, Dr. Mohanty worked to develop assays that would improve the care of patients beyond his reach. The tools we develop promote shared decision-making, much in the same way that a skillful physician in charge, like Dr. Mohanty, would engage trainees at a teaching hospital. These tools are designed to be easy to use and not to add to a busy clinician's workload. And one simply cannot accomplish this without taking the time to build empathy and compassion for those with whom they work, two qualities that Dr. Mohanty emphasized throughout his career. The ability to provide not only the right information, but the right amount of information requires one to tap into unique sources of creativity. And we can see the immense creativity that Dr. Mohanty possessed and expressed through poems, novels, stories, and plays. And finally, we took unique care to conduct exhaustive testing to ensure the effective operation of these tools. Much in the same way that Dr. Mohanty took such a rigorous approach in his clinical and laboratory investigations. As you can see, there is much to be learned from observing the life and the practice of an esteemed colleague such as Dr. K Mohanty. And I hope this lecture gave you a different framework on how to think about how you might translate the knowledge that you hold so that others can have access to it. I thank you very much for your attention, and I'm happy to answer any questions that you may have.
If there is any questions from anyone.
Hello, Dr. Susan.
Hello.
I am Dr. Anand Srinivasan, a faculty in the Department of Pharmacology. The presentation was really nice and it was quite inspirational. So I just have, some technical doubts, like apart from the nonlinear modeling which you had used for population kinetics, was there any implementation of artificial intelligence algorithms in the background?
Yeah, that's a really great question. No, there was not. We would like to. We're actually looking at that for another software application that we're building, sort of a Bayesian approach, but not maybe artificial intelligence in the way that you're thinking about it. We didn't have the capacity at the time to do that. We had some ideas of things that we would like to do from an artificial intelligence standpoint, especially as it related to optimization of these mathematical models. But that proved to be beyond the technical capability of the team that we were working with. And so we have tabled those ideas for now, but I can certainly see a number of applications for it as well. And man, that would really expand the application of these tools if we were able to do that. It's a great question.
Thank you. Especially that busulfan was, it was really inspiring. I mean, I really like that part. Thank you.
Thank you. Yeah, it's a bit of a challenge. You know, not everybody, not every patient's data fit the same type of model. And, you know, if you look at traditional pharmacokinetic software, it requires you to tell what model should be fit. Now, that's not a problem for my esteemed colleagues that are listening in today, but it is a problem for my physicians who don't have a pharmacology background. And so creating the algorithm in the background that really discovered on its own what the best model was quite a challenge, but made it far simpler to execute at the level of the provider.
Yeah, of course. Yeah, we can definitely appreciate that background efforts.
So I have a question. Since you are working on only drugs with narrow therapeutic index drugs, especially in the pediatric population, have you extrapolated the same in adults?
Yes, so when I talked about the additional revenue stream for the organization, those are actually for our adult hospitals. So the nice thing about that is now that we have this technology in place here, we can actually bring additional revenue by consulting for those adult hospitals as well. So we do all the testing with both pediatric and adult profiles. I mentioned we might run our software anywhere from 10 to 30 simulated profiles, and they span age, they span weight, they span gender, body composition. So depending on the drug and the variables that influence disposition or action, we will design our tests accordingly. We're building a much bigger software tool right now that deals with mental health. 92 different medications for adults and children. And again, we take all of those variables that are significant in their influence of dose exposure response and build them into the software.
Will the algorithm also design to develop the match the progress of the disease and the dose adjustment? How can you cope up with this? The disease will progress, it may have a downhill course, and the drug requirement may change with time. How can you adjust to that?
Yeah, that's a really interesting question, because unless you have a pre-existing disease progression model, it becomes a bit challenging. So busulfan, we're very lucky, right? Hopefully we're using it once for a bone marrow transplant, and if we're lucky, we never have to do it again. That patient will have accepted the transplant. The hemophilia obviously is a lifelong disease, and there the providers will typically repeat the testing. So repeat this type of testing that allows them to individualize as the child grows, or when there's a clinical indication that the dose is no longer working. So the child is presenting with more bruising and bleeding, that type of thing. You are perfectly correct that in an ideal circumstance, we would have a way to integrate it with the disease progression model and allow them to estimate, over a long period of time. The one thing that we did do with the hemophilia when we built in that population PK approach as well was to at least allow them to bring the patient back for just a single estimate and see how well they were doing over time. The goal would be for a software like this to perhaps assimilate that data and start to generate some of those disease progression models. But we don't have too many disease progression models in pediatrics. And even in adults, I think they're few and far between.
So there is not a lot of work need to be done. Yeah.
Yes, but very critical data. And man, if folks are interested in that field, I mean, it's ripe for application to the management of patients. It really is. I mean, even those data are useful in drug discovery, drug development. I mean, there's really a lot of value to disease progression modeling.
Right, because a lot of factors come into selecting a dose, like age, sex, then disease, concomitant medication, renal function, liver function, cardiac function. So a lot of things will decide how to choose a disease. And some patients have deficient in having some enzymes that does not interact with the drug. There may be overdose or therapeutic failure. So a lot of factors need to be considered. And this is what you have made. So we will like to interact with you more in future. Thank you. Now it is time.
Just any more questions from the audience?
Dr. Anand here again. I just wanted to know if it's not a secret, what program was used in the back end to run the nonlinear models.
Oh, we wrote the code ourselves. So we wrote the code, and I mentioned the refactoring, the multiple refactoring. We started in Perl, then changed to C++, then finally, then we had to change again to C# because that's the only thing at the time that could integrate with our EHR. Now with FHIR applications and the ability to talk to the EHR through an API, we're able to code using other languages as well. But we write all that code because the algorithms get to be very complex when you're requiring the software to do decision-making that the pharmacologist would normally do.
Thank you.
Thank you, Professor Susan, for such a wonderful talk. Now I request our head of the department, Professor Devashish Hota, to award the citation to Professor Susan. AIIMS Bhubaneswar takes the privilege of honoring you with Dr. K Mohanty Memorial Oration Award 2021 in recognition of your seminal contribution to the field of clinical pharmacology and medicine. So please accept from our side.
Yeah, I call on my colleagues, Dr. K Mohan.
It is my distinct pleasure. Thank you so very much. Just to be associated with Dr. Mohanty is a distinct pleasure.
Thank you.
So my colleague, Dr. Ritupurna, who is just next to me, and Dr. K Mohan next to Ritupurna. So we are all part of the Department of Pharmacology and Clinical Pharmacology here. So nice meeting you too, and we hope we can interact in future and more interactions in future. Thank you.
I hope so. I hope so. Thank you so very much.
Thank you.
Now I request Dr. Deepika Mohanty to present the Oration Award to Professor Susan. Video. Dr. Deepika Mohanty, please.
Yeah, I have the wonderful lecture, and I will start telling a few words about this lecture. Good morning, also good afternoon, and good evening to all attendees logged on to this J.K. Mohanty oration in Clinical Pharmacology 2021 from different parts of the world. I would like to thank the Director, All India Institute of Medical Science, Bhubaneswar, Dr. Gitanjali, and specifically the Department of Pharmacology for hosting this second Dr. J.K. Mohanty Memorial Oration 2021. As the daughter of late Dr. J.K. Mohanty, I express my deep sense of gratitude to Professor Susan, Director Healthcare Innovation in School of Medicine of the University of Missouri, Kansas City, for making time amid her busy schedule, as you saw, and delivering the second Dr. J.K. Mohanty Memorial Oration 2021. I know it is rather late in the day for her, getting close to midnight. So I won't take much time to put a few words for her. I'll only add a few words as a doctor, as a clinical hematologist, and also the daughter of a clinical pharmacologist. Thank you very much for your kind words for my father. A frontier science like medical science has been dramatic changes in the last 50 years. And we'll see more pronounced changes in the foreseeable future. Artificial intelligence and machine learning are hot topics for healthcare today. Yet, using AI ML models for real improvement in patient care remains difficult. Speaking of the developing world like India, the last few decades have seen rapid strides in non-invasive diagnostics, not to speak of the advancement of various technologies in surgical and therapeutic protocols. Notwithstanding the glory we have achieved in the treatment protocol and technological advancement, I must not forget that at the heart of it all is the patient and his wellness. Closely associated with it is the doctor-patient equation. This is a vital component of the equation. Doubtless, the diagnostic tools can and should supplement the clinical skill sets of the clinician, but they can't seek to replace fully, especially in a stochastic field like medicine that deals with animate humans. Because from them flow the soft skills, as you have already heard, like patient care that establish and strengthen the doctor-patient connection. The gleam in the patient's eye when he sees his doctor is one of the most reinforcing forces of the treatment protocol. It is not just a matter of mutual trust. It goes far beyond, like forces of the treatment protocol it is not just a matter of the mutual trust, it goes far beyond. It is psychogenic and psychosomatic. It helps as much in improved healing of the patient as it does in opening of the doors of the possibilities for the doctor to undertake the right treatment with a better understanding of his patient and a consequential holistic twist to his recipe and approach. Today's information technology is an enabler in multiple ways. To the patient, it offers a better understanding of the issue. This grasp, I guess, is relevant in pharmacogenetics and pharmacokinetics and pharmacodynamics and newly added horizon social pharmacology. Their main role is to improve patient care through the safe, economic, and effective use of medicines. Other variables, many in number, I admit, do also matter. Yet, there is one variable called empathy that does have a universal application and remains unvarying across the globe. I would suggest this should not be lost sight of. This I have learned from my father, who speaks often of bedside manners, good history taking, good clinical examination, and communication skill to convey the message clearly to the patient. Clinical pharmacology has been termed a bridging discipline because it combines elements of classical pharmacology and practice of medicine through innovative research, development, and also regulation of medications. I once more thank Prof Susan Abdel-Rahman for graciously accepting this invitation and for delivering the Dr. J K Mohanty Memorial Oration 2021. This I'm fortunate as a customary to hand over this memento to her, which is a silver plaque. Is what it is for her. It will reach her after some time, not immediately, because this is a presentation. And I would like to thank, lastly but not the least, Dr. Devashish Hota, head of the Department of Pharmacology, and Professor Dr. Rituparna Maiti for their untiring effort to make this possible. My only regret is that we missed the presence of our beloved mother, Remalata, who we lost earlier this year. Thank you to one and all. Good night and also good day to all others. Dr. Maiti, I finished my few words. I hope I am not lengthening it. But one thing I would like to, I couldn't ask the question, I would like to tell Dr. Susan that being a hematologist, I can give some example that we found that the antiplatelet drugs has much to do with the that is the ethnic variation is there. So also the anticoagulant. Some work of ours is also published that we have to reduce the role of the anticoagulants, especially vitamin K antagonists. So these are some of the work which we have involved, and it really needs the population genetics or pharmacogenetics for this point. It's very, very important. Very nice to hear your lecture. It is very much in line with my father's ideal. Thank you.
Thank you,
Thank you so
Deepika
very
ma'am.
much.
Now I request Dr. Anand Srinivasan, Associate Professor in Department of Pharmacology, to conclude this program with vote of thanks. Please.
Good morning to all. So this oration is about paying tribute to the late Professor Dr. K Mohanty and the Department of Pharmacology and AIIMS Bhubaneswar. Thank Professor Susan Rahman for displaying her work and guide the pharmacologists like what can be done in the future, and especially since the work has actually is aligning with our future goals also, we found it very interesting. Thank you, Professor Susan. And I thank Dr. Deepika Mohanty for taking this initiative to propagate the knowledge of pharmacology among the various pharmacologists in the country. I also thank Professor Geetanjali Padmanabhan, the director of AIIMS Bhubaneswar, in helping us in organizing this oration, and she only suggested Professor Susan's name, and definitely we are lucky to listen to her today. Also thank Dr. Debashish Hota, Dr. Rituparna Maiti, who are professors in the Department of Pharmacology, and I also thank Dr. Vishumo Anpadi, Dr. Monalisa Jana, and Dr. Vikas Ranjan Meher, who are my colleagues in the Department of Pharmacology in organizing this workshop. I also thank our IT cell in enabling the logistics for this presentation. And lastly, this program would not have been a success if not being supported by the various participants who have shown their interest and who have stayed until the end. Thank you all.
Thank you all.
Thank you, Susan. Thank you all participants. Thank you, Deepika ma'am. Bye-bye and have a good night, good day, and good night to Susan. Good night.